So today we'll be discussing on the etiology of cancer. So what are the causes of cancer? We will discuss in the detail. So there is a different factors that is known as the predisposing factors like age, hereditary, environmental factors, and acquired precancerous disorders. So this also affects the cancer. And the agents, that is a carcinogenic agents that lead to the cancer, like you can say the physical, chemical, and the biological agents. So in the physical, it is a radiation, radiant energy, which causes the cancer. The different chemicals that also lead to the different type of cancer, and the biological agents, that is a virus. We can say it is a oncogenic virus. Either it might be DNA or RNA. Both can act as oncogenic virus. So we'll discuss one by one all the predisposing factors and the carcinogenic agents. The first one is the age. So this cancer can develop in the any age. Okay, but mostly you can see commonly in the age group more than 55 years. It can develop in any age, but mostly in the or very common in the age group above the 55 years of age group. There is a few cancer which can be seen in the children, which is common in the children less than 15 years of age group. Example, retinoblastomas, neuroblastoma, Wilms tumors, lymphoma, leukemia, and sarcoma of bone and skeletal muscles. So this retinoblastoma, it is a rare form of cancer that rapidly develops from the immature cell of retina. So it is related to the retina. Okay, rare form of cancer that rapidly develops from the immature cell of retina. That is a retinoblastoma. Neuroblastoma, it is related to the neurons. Okay, it develops from the immature nerve cells. It is a cancer which is developed from the immature nerve cell, that is a neuroblastoma. Wilms tumors, it is related to the cancer of kidney. The cancer of kidney. Lymphoma and the leukemia, these are the blood cancer, you can say. Blood cancer. Okay, this lymphoma, it is a cancer of the lymphocytes, which is a blood cells. Blood cells. Lymphoma. This is due to the increase in the number of lymphocytes, and the leukemia is a blood cancer. Sarcoma, it is a cancer of the connective tissue. Sarcoma is a cancer of connective tissue. So these are the few examples which is common in the children below 15 years of age group. But if you'll see as such, cancer can see in the any age group, but in the adult, most common in the age group more than 55 years of age. This is one predisposing factor, that is the age. Second one is the hereditary, which play important role in the carcinogenesis. If your parents, our parents having the some type of cancer, it is transferred to the offspring. Okay, you can take the example retinoblastomas and the chromosomal DNA instability. This retinoblastoma inherited a autosomal dominant trait, and 40% of this retinoblastoma comes from the family. Okay, it is a come from the family, around the 40%. Out of 100%, if you will take the retinoblastoma, out of that 40% is a from the family you can get. It can be inherited from the family. Chromosomal DNA instability, it is a autosomal recessive trait, and conditions are characterized by defect in the DNA repair. Already you have studied the DNA repair, different mechanism, how DNA is repaired. So there will be defect in the DNA repair mechanisms. So this is second predisposing factors, that is heredity, which is come from the family. The third one is the environmental factors. More than 80% of the human cancer is caused by environmental factors. This is the more predisposing factors which help in the development of cancer. Example, you can tell lifestyle, dietary habits, occupations. Okay, these are some of the examples of the environmental factors like the lifestyle, cigarette smoking, tobacco chewing. These are our habit during the lifestyles, which contributing factor for the development of cancer. Dietary, you can see groundnuts and some foodstuffs. When it is get infected with the fungus known as aspergillus that produces the alpha toxin B1, which is carcinogenic. Alpha toxin B1, it is a carcinogenic, which is produced by the fungus known as aspergillus. When this alpha toxin get contaminated with groundnuts and the foodstuffs, then person will develop the cancer. Like occupation, asbestos, which is used for the protection during the fire. Fire protective materials made from the asbestos, which is white and very flexible. This substance also can causes the cancer, like benzene, naphthalamines, berylliums. So these substances causes the cancer. One more you can see the iatrogenic, certain therapeutic drugs. This is due to the physician while treating the patient with the help of some drugs that may causes the carcinogenicity. The drugs can causes the cancer. So that is during the time of treatment by the physicians. Some of the drugs might causes the might have the carcinogenic effects. So these are the environmental factors which might lead to the cancer. Like our lifestyle, which is very common in most of the age group. Some of them uses a style of smoking. Somebody may have the habit of taking the smoking and the tobacco, and that is the cause for the cancer. Many cancer you can say. The fourth one predisposing factor is the acquired precancerous disorders. Like some of the disorders which is developed in the patient that might lead to the development of different cancers, like leukoplakia of the oral mucosa and genital mucosa. So this is a white patches. Leukoplakia means the white patches of oral mucosa and the genital mucosa that might develop the carcinoma known as squamous cell carcinoma. Okay, so this is a one acquired precancerous disorders, leukoplakia, that is a white patches, and finally it leads to the cancer, the squamous cell carcinoma. Cirrhosis of liver, which mainly you can see in the case of alcoholic persons or persons suffering from the different viral diseases. Okay, cirrhosis of liver. This also may develop into the hepatocellular carcinoma. This is not the cancer. Cirrhosis of liver is not a cancer, but later on that might causes the hepatocellular carcinoma. Ulcerative colitis. This also can produces the adenocarcinoma of the colon. This ulcerative colitis, it is inflamed large intestine, and you can see the rectum. Large intestine and the rectum are inflamed. That is a ulcerative colitis that might also produce the cancer that is known as adenocarcinoma of the colon. So these are the some conditions which might causes the development of different cancer. So that was the predisposing factors. Now come to the carcinogenic agents that causes the cancer. So it is divided into the three groups: chemical carcinogen, physical, that is the ionizing radiation, so you can say the radiant energy; biological, that is the oncogenic virus, either it might be DNA or RNA, which act as a carcinogen and causes the different type of cancers. So we'll discuss one by one: chemical carcinogen and ionizing radiations, how it causes the cancers, and this virus, what is the mechanism to develop the cancer. So these chemical carcinogens are the chemical compounds that can produce the cancer, which produces the cancer. Around, already I have told, 80% of the human cancer is caused by the chemical carcinogen. And this chemical carcinogen can enter into the human body through the many mechanisms, like during their occupations, through the diet, as a drugs, or during our lifestyle. So occupation like asbestos, which is working in the fire office. Okay. In the diet, alpha toxin, which can be get contaminated with our food, produced by the fungus known as aspergillus. And drugs, diethyl stilbestol. This is the synthetic estrogen which is used to prevent the miscarriage. This is used to prevent the miscarriage. So this might also causes the cancer. Lifestyle, as I told, smoking, tobacco use. This is our lifestyle. Through all this route, the chemical can enter into our body and cause the different changes and finally lead to the cancer. So there are two types of chemical carcinogen: direct acting and indirect acting carcinogen, chemical carcinogen. So this direct acting chemical carcinogen do not require any metabolic activations. Metabolic activation is not required and directly react with the nucleophilic group in the nucleic acids. Nucleic acid, you have the two types, DNA and RNA, and nucleophilic groups. That is a phosphate group. It can go and bind to the phosphate group of nucleic acids. Example, alkylating agents and acylating agents. So these acylating agents transfer or donates the acyl group. And these agents go and bind to the nucleophilic group of nucleic acids. That means phosphate group of DNA and RNA can go and bind there, and it will act directly. Second group, indirect acting carcinogens. This is also known as the pro-carcinogen. This requires the metabolic activations to become the carcinogenic. Okay. To become a carcinogenic, it requires the metabolic activations. Example, you can see the polycyclic aromatic hydrocarbon. Example, benzopyrene and alpha toxin B1 and aromatic amines. So this requires the metabolic activations, then only it can be carcinogenic. Okay, that's why it is known as the indirect acting carcinogens. It does not act directly. It requires the metabolic activations to become the carcinogenic. This is a mechanism of action in the both the group, how it develops into the cancer. Direct acting carcinogen, no metabolic activations, reactive electrophiles interact with the neutrophils. Okay, nucleophiles interact with the nucleophiles, that is our phosphate group. Mutation in the DNA, finally it's developed the cancer. Second group, indirect acting carcinogens, requires the metabolic activations and the same pathway. It can bind to the DNA, mutation in the DNA, and finally there is development of cancer. So this is the way how the chemical carcinogens can cause the cancers. It causes the mutation in the DNA by binding to the DNA. Okay. So these are some examples of the chemical carcinogens. Polycyclic aromatic hydrocarbon like benzene, benzopyrene, aromatic amines, nitrosamine, and various drugs, alkylating agents, you can say, naturally occurring compounds, alpha toxin B, actinomycin D, inorganic compounds. Okay, inorganic like arsenic, asbestos, beryllium, cadmium, chromium. So these are some of the chemical compounds which is a carcinogenic. So this, if you take all these chemicals, it develops the cancer in our body. So there's some example for chemical carcinogens which is related to the cancer, which develop into the cancers, which causes the cancer. So this DNA damage caused by chemical carcinogen, it causes the mutation in the DNA. So what is the DNA damage caused by the chemical carcinogen? So this chemical carcinogen damage DNA in a variety of way, inserting or removing the individual base. Just note down, altering or removing the individual base, triggering the break in one or both the strand. So either it can remove the individual base or alter the individual base, or you can see in the diagram here. This is one strand, this is another strand. Okay, there is a cut in the single strand. This is a DNA damage, or cut in the both the strand, double strand cut, either single strand cut or double strand cut. This is the DNA damage. Another, you can see this base. This base has to be attached to opposite one. Here, attached here, attached here, but it's attached to the adjacent base. This is also the DNA damage. Cross link between the DNA strand. See, like this. Hydroxyl group is added to the base. This is also the DNA damage. Here you can see the base is missing. Base is missing. Okay, so this is a different type of DNA damage that is caused by the chemical carcinogen. You can note down single strand break, double strand break, link between the adjacent base, cross link between the DNA strand, hydroxyl group is attached to the base, missing base. Okay, these are the DNA damage caused by the chemical carcinogen. So for this, you can perform one test, either these chemicals are carcinogenic or not. How to find out? How to know the carcinogenicity of the particular chemicals? So for that, you can perform one test that is known as the MS test, that is used to detect the mutagenicity of the chemical compounds. So they have done, you can see here. So chemicals causes the reversion mutation in the bacteria, so it can grow without the histidine. So how you can test this one? So you can take the bacteria that is known as the S. typhimurium. This is Salmonella typhimurium, which does not contain the histidine, which is not able to synthesize the histidine. Okay, histidine is not synthesized by the Salmonella typhimurium. Okay, then you can take the extract from the mice. Now this mixture are plated on the plate which is lacking the histidine. This is agar plate. Okay, this is agar plate which does not contain this histidine. In the nutrition, we will add the nutrition without the histidine, and this test chemical which is added to the filter paper. Okay, this is the filter paper. From the filter paper you can make like a disc. Okay, this contains your chemicals. You can take the filter paper, in that you put the chemicals and dry it. Now you can place on the agar plate. Okay, place the on the agar plate. First, this agar or this plate which does not contain the histidine, then pour this mixtures, Salmonella typhimurium, okay, and the extract from the mice plated here. Okay, in the normal condition, what will happen? The cell, this bacteria will not grow. See, very few colony you can see. Bacteria will not grow because for the growth of this Salmonella typhimurium, it requires the histidine. If this chemical is not the carcinogenic, if it is not the carcinogenic, this bacteria will not grow. Okay, bacteria will not grow because this medium does not contain the histidine. If the histidine is not present, this bacteria will not grow. For the growth of the Salmonella typhimurium, it requires the histidine. So if this chemical does not having the carcinogenic effect, this bacteria will not grow. If this chemical is carcinogenic, so what will happen? There is a mutation in this bacteria. Mutation. Okay, that is known as the reverse mutation. So now this bacteria can produce the histidine due to this chemical, which is the carcinogenic. the chemical which is a carcinogenic, which causes the mutation in this bacteria. There is a reverse mutations. Now this mutated bacteria can synthesize the histidine. It synthesizes the histidine. Due to that, you can see here more growth of this Salmonella typhimurium is grown here. So this indicates that this chemical is a carcinogenic. If this chemical is not the carcinogenic, it will not cause the mutations. So unmuted bacteria cannot synthesize the histidine. So it will be like just like this. See, it's not near to the chemical. See, 1 or 2 colony only you can see. But if it is carcinogenic, that chemical is carcinogenic, it's causing the mutation in the bacteria, which is capable of synthesizing the histidine, and the bacteria will grow like this. So based on this, you can say by this test, this chemical or this chemical compound is carcinogenic. Okay, this is a very simple test, and easily you can find out it is carcinogenic or not by using the Salmonella typhimurium, which is not able to produce the histidine. So this is another agent which causes the cancer or carcinogenic. Radiant energy. So it is a UV rays, X-ray, and the gamma rays having the mutagenic and the carcinogenic effects. These rays, UV rays, X-ray, and the gamma rays are mutagenic and the carcinogenic. So these rays damage the DNA in the several ways. There is a many way to damage the DNA. Like UV rays result in the formation of the pyrimidine dimers. If you remember the purine and pyrimidine base. So pyrimidine base are uracil, thymine, and cytidine. So pyrimidine dimers, mainly you can see the thymine. Two thymine will combine together to form the dimers. That is a pyrimidine dimers. Thymine, okay, pyrimidine dimers. So this is also the causes the DNA damage. That is result by the UV rays. Another, you can say the X-ray and the gamma rays produces the point mutations and the chromosomal break. This chromosomal break can lead to deletion of large portion of DNA. Okay. UV rays causes the pyrimidine dimers, but the X-ray and the gamma rays causes point mutations and the chromosomal break. Due to this chromosomal break, the large portion of DNA can be deleted. Large portion of DNA can be deleted. So that is the DNA damage. Okay. This is the one way to damage the DNA. Second way, this X-ray and gamma rays also causes the formation of the free radicals. There is a formation of the free radicals in the tissue which interact with the DNA and other micromolecules, leading to the molecular damage. If it is a combined with the DNA, causes the DNA damage. The free radical which is produced in the tissue due to X-ray and the gamma rays. Okay. So the damage in the DNA is believed to be causes for carcinogenicity with radiant energy. So these three, UV rays, gamma rays, and the X-ray. So UV rays only related to the dimer formations, but this X-ray and the gamma rays, point mutation, chromosomal break, and the formation of the free radicals. And finally causes the DNA damage, which might be the cause for the carcinogenicity. So this is the mechanism of radiant energy, how it is related to the carcinogenicity. The third agent is the biological agent, that is the oncogenic virus. You can see the few example of DNA virus. So DNA virus, example the papavirus, herpes, adeno, pox, okay, hepadernavirus. These are the member of that DNA virus. Human papilloma virus, Epstein-Barr virus, hepatitis B virus. So these are the DNA virus, which is oncogenic virus. And the RNA, acute transforming virus, raw sarcoma virus, leukemia sarcoma virus, slow transforming virus, mouse memory tumor virus. So these are the few examples of the oncogenic virus, which is DNA and RNA virus. HIV, human immunodeficiency virus, HIV-1 and HIV-2 both. This also comes under the oncogenic virus. So what is the mechanism of carcinogenesis or cancer development due to this virus? How this virus can causes the development of cancer? Yesterday in class we have discussed different type of gene like oncogene, proto-oncogene, tumor suppressor gene. So this tumor virus having the gene known as oncogene. It carries the oncogene genetic elements, that is an oncogene in this tumor virus that can integrate into the host genome. This oncogene, which is present in the virus, integrate into the host genome and encodes the viral oncoproteins. Just note down, encodes the viral oncoproteins. This viral oncoprotein forms the complex with and bind to the tumor suppressor. The function of tumor suppressor is to block the abnormal growth and the malignant transformation. This tumor suppressor is block the abnormal growth and formation. If this tumor suppressor is bind with this viral oncoproteins, so it will not function normally. The normal function is to block the abnormal growth and malignant transformation. But if once it is bind to the viral oncoprotein, it is not do this function. It is not able to do this function. Okay. So due to that, there is a proliferations and finally development of the cancer. So this tumor suppressor not able to block the abnormal growth because it is already bound to the oncoprotein, viral oncoproteins. So there is abnormal growth, proliferations, and finally development of cancer. So totally depends on the oncogene and the viral oncoproteins. So this oncogene integrated into the host genome and encode the viral oncoproteins, which combine or complex with the tumor suppressors. So these tumor suppressors are not able to work normally. That leads to abnormal growth, proliferations, and development into the cancer. So this is the mechanism how this virus, oncogenic virus, causes the cancer. So this is the molecular basis of cancer by all these three: chemicals, radiation, and the viruses. So this is the normal cell. If you remember the replications, so during the replications, there will be the DNA damage. Okay, that is a normal, which can be again repaired by the different enzymes. Different type of DNA polymerase will come and remove that damage. But the damage caused by this chemical, radiation, and the virus, if it is repaired itself with the help of the p53, okay, p53, that will stop the cell cycles and try to maintain the DNA damage. If that will successfully done, no problem. Okay. If it is failed to repair the DNA, there is a mutation in the genome of the somatic cells that causes the mutation in genome causes the activation of growth promoting oncogene. Second one, inactivation of tumor suppressor gene. Third one, alteration in the gene that regulates the apoptosis. Thus you remember, if there is DNA is not repaired, so that causes the mutation in the genome of somatic cells. And due to that, there is the activation of growth promoting oncogene, inactivation of tumor suppressor gene, and alteration in gene that regulates the apoptosis. So what will happen after these three sequence will takes place. There is up-regulated cell proliferations. Tumor suppressor is inactivated, and oncogene growth promoting oncogene is activated. So there is up-regulated cell proliferations, clonal expansion, tumor progressions, and you'll have the cancer. Finally, will develop the cancer. Alternation in the apoptosis, decrease the apoptosis, same thing, clonal expansions, tumor progressions, and you'll have the cancer, which can travel to the one part to another, that is with metastasis, invasion into the blood and reach to some other part, that is a metastasis. So this apoptosis also used to remove the cell which contains the DNA damage. If this is also decreased, there will be clonal expansion. There is a proliferations and tumor will be formed. Even with different factors which is also supporting here. See, tumor progressions is supported by the angiogenesis, escape from the immunity, additional mutations. So all these factors will contribute for the tumor progressions. Angiogenesis is a more blood supply. More blood vessels will be formed which supply this blood for the tumor progressions. So these are the factors which also help for the tumor progressions. Additional mutation will take place, escape from the immunity and angiogenesis. More blood vessels will form to supply blood and which help in the tumor progressions. So this is a mechanism how from these agents, chemical agents, physical agents and biological agents causes the cancer. So this is just one example you can see. Normal epithelium, then hyperproliferative epithelium, early adenoma, intermediate adenoma, late adenoma, carcinoma, and metastasis. So this is the, you can say, the steps in the formation of the metastasis. Metastasis is the last part you can say, which can transfer to the different other part of the body. Malignant tumor you can say. So this is how it will be reached to the other part of the body. Steps in the process of metastasis. This is the primary tumor in one part you can see here. Abnormal growth of the cell. See, abnormal growth, which causes the invasion in the blood. Okay, invasion in the blood, and you can see here it is travel into the blood. Now extra invasions, so from the blood again it came out and causes the cancer to some other part. That is the secondary tumor. This is the primary tumor, which is the site where it is formed at the first. Then it will travel through the blood, invade the blood, then it reaches to some other organs and causes the secondary tumor, causes the cancer that is known as the secondary tumor. So you can see this is the extra angiogenesis. Angiogenesis, this one, it is formed for the blood supply. More blood vessel is formed for the blood supply of these cancerous cells. Here also you can see additional blood supply. This is angiogenesis. Okay. So this is how the cancer travel from one organ to another organ, and this is known as the metastasis. So there is many changes, biochemical changes you can see during or inside the tumor cells. In the growing tumor cells, there is many biochemical changes you can see. Increase the activity of ribonucleotide reductase. If you remember the function of this ribonucleotide reductase. So what will do, the ribonucleotide, it is converted into the deoxyribonucleotide. That means directly help in the DNA synthesis. Increases the DNA synthesis you can say. This ribonucleotide converts ribonucleotide into the deoxyribonucleotide, which is required for the synthesis of DNA. So it is a activity of this enzyme is increased. That means there is increase in the synthesis of DNA as well as RNA. Okay. Decrease the catabolism of pyrimidine. Pyrimidine catabolism will decrease. That's why it may forms the dimers, pyrimidine dimers. Increase the rate of glycolysis, aerobic glycolysis and anaerobic glycolysis. Glycolysis means the glucose is break down into pyruvate or lactate. Aerobic conditions it forms the pyruvate. In anaerobic condition it forms the lactate. Okay, so this will provide you the energy. Energy is provided from the glycolysis. So more rate of glycolysis takes place in growing tumor cells to provide the more amount of energy, more ATP, to supply the more ATP. So this glucose is converted into the pyruvate or lactate. If there is oxygen, it forms the pyruvate by the aerobic glycolysis. And if there is no oxygen supply through the anaerobic glycolysis, it forms the lactate. Okay. Another is alteration of isoenzymes profile, like different fetal pattern you can see. Even the synthesis of the fetal proteins like CEA, carcinoembryonic antigens, that is also synthesized in the tumor cells. So you can measure the CEA in the cancer patients. You can measure the CEA. This is a, you can say, the tumor marker for the diagnosis of the tumor, because it is synthesized as a fetal protein CEA, carcinoembryonic antigens. So you can remember, if you forgot other things, remember the increased synthesis of the DNA and RNA, and rate of glycolysis is increased. Not only one glycolysis, both aerobic glycolysis as well as anaerobic glycolysis. The rate is increased to supply the energy. The other changes which can be seen in the growing tumor cell, loss of differentiated biochemical functions like diminished synthesis of the specialized proteins. So many protein is not formed in the growing tumor cells. Inappropriate synthesis of certain growth factors and hormones, which can support the tumor. Inappropriate synthesis of the growth factors, which supports the tumor. Alteration of the cell surface due to changes in the composition of glycoproteins and the glycosphingolipids. Glycoproteins and glycosphingolipids. Secretion of angiogenesis factors that stimulate the development of blood vessels to support the tumor, because it requires the continuous blood supply to the tumor cells. So there is a different angiogenesis factors which can stimulate the development of the blood vessels. Production of the matrix metalloproteases, collagenases that facilitate the invasion of the adjacent tissue. Okay. So these are some of the changes that you can see in the growing tumor cells. So some biochemical and genetic changes occurring in the human cancer cells you can see here. Okay, in one diagram everything is just mentioned. Increased anaerobic glycolysis, degradation of the certain enzymes, release of the enzymes, okay, release of the angiogenic factors, biomarkers, ability to metastasis, decrease the addition of the neighboring cells. So everything you can see here in the one diagram, everything is mentioned. Mutations, abnormal cell cycles, chromosomal abnormalities, activation of oncogenesis, inactivation of tumor suppressor gene, alteration of small RNA molecules, so all this. Okay. Biochemical, this size, and genetic factor is this size. Okay. Genetic changes you can see here, and biochemical changes on the left sides, and the right side it is a genetic changes. So these are the some of the MCQ. Before that, you can summarize the today class. We have discussed regarding the etiology of cancer, in that predisposing factors and carcinogenic agents like chemical carcinogen, radiation, how it causes the cancer, and biological agent, that is oncogenic virus, different type of virus which causes the cancer, and how to test the carcinogenicity of different chemicals by using the MCOMS test. So this is all regarding the etiology of cancer. So there is a few MCQ through that we can go. Name the cell which lost their control of the regulated divisions, differentiation, and apoptosis, and that is known as which of the flowing mutation causing the Burkitt lymphoma. We have discussed in the last class. Out of this 4, 1. Okay. So find out which one, and you can tell the answer in the next class. In the tomorrow class, I will ask the answer of these questions. If the DNA is damaged, which of the following gene arrests the cell cycle? RBG, P53, okay, which one? Carcinogen, what do you mean by the carcinogen? Out of these 4, which one? Okay, so these are 4 MCQ. Tomorrow, just I will ask the answer of this MCQ. So any doubt? So this is the end of the class. If you have doubt, you can ask me. Any doubt? Do you have any doubt?
Excuse me, sir.
Yes.
Sir, can you